often have to juggle a lot of different projects and experiments, and it can get really overwhelming unless you organize. And so, if you set good habits early on in terms of things like how you name your files and how you organize your notes, this is going to help you stay cool, calm, and allow you to carry on all those cool experiments. So, this is just what works for me, how I like to organize my things personally. It might not be what works for you, so just find what works for you, because that's the most important thing, is to just be consistent, and to establish it early on so that you can then know where to find things, know how things are labeled, be able to be really organized and under control. I found that it's really helpful to save the date in your file names. And so, more on this in yesterday's post, but it makes it really easy to cross reference things. Speaking of, when you're saving those files, don't include spaces in your file names. Instead, use underscores so you don't confuse the computers. Having the date saved is going to be helpful so that you can kind of do version control. You can also use like Git and GitHub and things like this. Basically, you want to have multiple versions of something when you're working on a paper, say, and you're making a bunch of edits. Maybe you wrote something and then you cut it out, but then you realize you wanted it after all. And so then you can go back to an earlier draft. And so what I do is I put the date in my file names, and I'll have multiple versions of it, all with a different underscore date. And this allows me then to go back and revive things and revisit things and various things like that. Speaking of saving, though, whatever your strategy, make sure you save early and often. So save to the cloud. Don't wait to learn the hard way and have all your stuff be um your computer be lost or broken, and then you are missing everything. Having it on the cloud also makes it so that you can access it from, say, work if you don't have your laptop with you. Also, like to regularly back up my laptop to an external drive just in case. And so this way, if anything happens to my laptop, I can then recreate it easily. Okay. So one of the most important things is to keep detailed daily lab notes and organize by date, going back to that key importance of the date for cross-referencing. I like to keep a physical notebook and a electronic notebook. I actually take notes in my physical notebook, and then I take pictures of those notes, and I actually stick them into a folder on my computer, and I also stick them into the pages of my digital notebook. But sometimes when you're in the lab and you're doing experiments, I like to write things down. And when I'm all goopy and messy, but then I want to have it in a searchable format. And so this is good with it. So the electronic version is good for that. I use OneNote. There are different tools that you can use. I know a lot of people use like Benchling and like that. But I use OneNote and I organize things. I have a tab for each month, and then within each month, I have a tab for every day. And then I have some other things that I'll talk about, including like a results or figures tabs, to do cheat sheets with buffers, formulas, and things like this. But you want to, the most important part of that is going to be your daily notes. Make sure that you have the notes written before you, like, while they're fresh. And so it can be really easy at the end of an exhausting day to just want to go collapse, but make sure you've at least jotted down some notes while things are fresh, and then you can better analyze things once you've gone a little sleep, but you'll have all those details written down. And really important to like write down your impressions and various things like this that I go over in another post. In addition to those like daily notes, I have other things in my electronic notebook as well as in folders on my computer to keep it easily accessible, what the things that I need a lot, so that I don't have to go search for them. This includes like buffer recipes. So I keep buffer recipes. Some of them you can like write on post-it notes on your bench, which isn't the most searchable, but it's helpful for when you need them right away. I also keep buffer formulas in my notebook, as well as in a tab on like in my protocols and things like this on my computer. So lots of places where I have those buffer recipes. And then I'll show you where I keep spreadsheets where I can actually calculate master mixes and things like this to make how much I want of whatever volume I want based on the formula. It's good to keep accessible like important snippets of code for doing various things, various file paths and things like this, so you don't have to keep looking them up. This will just depend on whatever project you're doing, but you don't want to have to keep Googling, how do I add a column to my pandas data frame? And things like this. So it's helpful to have those in a place where in your notes where you can just easily search for them. I also keep like a cheat sheet of things that I often forget, like what voltage to run your Western blot at, or what OD to infect your cells or like harvest your cells and things like this. So various things, whatever the things that you often forget, just put them in that little cheat sheet. It'll help you. And then there's a couple I'll talk in more detail about. One is the experimental figures and one is the protocols. And so basically I make experimental figures as I go. Don't wait until you need them. And so I make these figures as I go. I'll show you in a second. And then I place the summary figures for related experiments in the same location for easy comparison. So what I'm talking about with these figures is basically kind of like an experimental figure that you would see in a paper with some extra information that you can then crop out strategically if you need to present this. This includes like your methods and experimental details, as well as the date. And having these figures all ready to go is going to be really helpful both for yourself as you're looking back to try to see what you did and compare results, as well as if you need to do a presentation or if your boss, your PI, like comes and asks you for some results, and then you'll have that ready to go. So you don't have to worry about going and finding and digging all the stuff and making this nice figure because you've already got it made. So I use Adobe Illustrator to do this. A free alternative is Inkscape. Use whatever works for you. As with all of this, just make sure that you save them all in a place where you can easily access them. Now let's talk about things that you can do to make your experimenting easier, starting with those protocols. So I write really detailed experimental protocols that I can easily access. And when I say detailed, I really mean detailed. So I really plan out, what do I need to pipette where, when, how am I going to order my samples, and things like this. When possible, I like to do my calculations and all of this the day before, and then look it over with fresh eyes in the morning and double check my calculations. For each experiment I do, there's going to be some stuff probably that's going to be unique to that specific instance of doing it. So what samples am I working with today? So that'll influence things like how much of the master mix do you need to make? Or what order are the lanes in your gel? But there's also things that are going to be constant, the basic of the experiment. And so I keep like a kind of generic version of the protocol that I can then basically annotate and edit for that specific instance of doing the experiment. And then I have this annotated copy that I can both work with in the lab. I like to print it out and like hang it up in front of me, as well as like write on it the details, like maybe your nano drop reading or the balance reading. If anything went off of script, you had to do anything differently, if you had any observations, all this stuff I write onto that sheet that I have with me. And then I can even just like take that into my notes, as well as take a picture of it and then annotate it back on the file. I have a version, I save it with like the date. There's the date again, so I'll save a version of the protocol with that day's date and any notes I added, which I'll add in like blue if it's just kind of notes, and red if something went wrong. So you don't want to see too much red in my protocols. But this way I can then see I have the protocol and I have information about how it went last time and things like this. And it can be really, really helpful, especially when you're trying to optimize a technique. So yeah, like I said before, I like to do things the day before at least, so I can have some time to really think it through. And sometimes you want to make sure that you actually thought things through correctly. I have more on planning experiments in another post, but you want to make sure that you know that you have like your controls and you have everything that you need. And that when you have things planned out really well, then you can focus on the experiment at hand. To help you with that planning, basically, I like to make spreadsheets for calculating my master mixes. So for example, if I have like an RNA gel extraction buffer and I know what the final components are and the final concentrations, but sometimes I want to make different volumes of it. Well, now I can use a spreadsheet to help me calculate out what I need to do. And so I have an example of this as well as examples of what I'm going to show you next, some of my inventories on my Google Drive. I have a public Google Drive with some resources, and I will take you there in a second and show you this, and hopefully it'll help you as well. But we're going to look also at inventories. So basically, I keep track of the different things I have, including primers and other oligo sequences, plasmids with information about their size and their antibiotic resistance. When relevant, if they're for like a recombinant protein expression, does that protein have a tag? What's the size and the PI of the protein with and without that tag? What is the status of my cell pellets if I'm expressing the protein and then I harvest the cells and I have these pellets of cells in the freezer? I want to know what I have. If I do I need to express more of it, or do I have plenty I can then go break open? This saves you from having to dig around in the -80 only to figure out, oh, well, I don't have any of those pellets, as well as like purified protein preps, etc. And so I will show you in a second where I have this on my Google Drive. But basically, during my PhD, I had over 140 protein contracts. So different proteins or versions of the same protein, and I gave them each number codes, and I had this really organized system in order to keep track of the cloning as well as the purification and things like this. So let's go take a look at that. So here's an example of a protein construct inventory. So you have like the vectors, and or your plasmids. Basically, I have like a nickname, its full name, description, tag, plasmid size, and insert size, size of the protein, various things like this, as well as notes about where it came from, or how I cloned it. I also have information about various proteases and where they cut, how big they are, as well as information about the sizes of the various tags that I sometimes use on my proteins. I also have an inventory. I also keep inventory. So like cell pellets, when I have pellets of cells that I express protein in, things like the growth and induction conditions if relevant, the media type, the cell type, the number of tubes. So you don't have to keep digging to see if there were 4 tubes or 3 tubes, because it'll be on the top. You'll know, or in your notes, you'll know how many tubes there were to be digging around for. And hopefully you don't have to dig around too much because you have your pellets organized well as well. Also for your purified protein, things like the name of the protein, the size, the prep date, mgs per mil, micromolar, the stored form, how many aliquots you have, what buffer is it in, various other notes on the protein. When you're actually wanting to work with protein, sometimes you want to make consistent amount, like load similar amounts on the gel. So I have like a spreadsheet where I can kind of easily calculate how much to load, as well as various master mix spreadsheets where I can then calculate based on the volume I want, how much of each different component do I need to add. And so I have some examples here of various things like this if in the uploads folder. That'll hopefully be helpful to you. And so, yeah, so I keep basically, when I have experiments where I have to make master mixes, I keep these spreadsheets for these different experiments, and then I can just kind of modify it to how many reactions I have or what final volume I want in order to know how much I need to make. And this makes things really, really easy. And then I can just like copy and paste this into the protocol, the annotation protocol, and then print it out and things like this. So really, really useful, at least for me. And so if you are interested in trying it out or doing something similar, basically you can go check out the uploads and then that'll link to, and you can try it out. So we've talked a lot about the day-to-day planning. So how to plan out ahead of time. If you have multiple things to do, plan how you can stagger, look for those long steps. And which in addition to like being used to get coffee, can be used to kind of stagger out different experiments and things like this. I also like to want to think, get you to think about longer term or bigger picture thinking. So periodically, every couple weeks or so, I make a to-do list and a project status summary. And so I have tabs for these in my electronic notebook. And so this will help me know where I am and what I still have to do. And when I talk about a to-do list, it's not like I try to sometimes schedule things out like a week at a time, so that I'll say, okay, on Monday, I'm planning to do this. On Tuesday, I'm planning to do this. On Wednesday, I'm planning to do this, blah, blah, blah. And these are the longer term goals I have. This is what I'm working towards. But it's really important to stay flexible because science doesn't always like to follow your schedule. Your experiments might not work. You have to redo them. Or you might be in the middle of your experiment and the machine might give you an error code. And so it takes you a lot longer than you thought. Also, meetings tend to magically materialize. So bottom line is try to be prepared, try to plan ahead, but also be flexible. This can be really hard when you're really set on doing an experiment or you have everything planned out really carefully and then you have to change things. But that's how it goes. And so don't try to not let that stress you out too much. In addition to staying on top of what you're doing, it's also really important to stay up to date on what others are doing. So there's literature and papers coming out all the time, and it's way too much for you to read it all. Even like to find it all, let alone read it all. But you can use tools to help it make it easier for you to find papers and things of interest when they come out. So you can set up alerts through like Google Scholar or PubCrawler or PubMed. You can set up alerts based on the subject or based on the author. You can set up alerts for whether when a paper is cited, and various things like this. And I go over this more in another post I can link to. I also use an RSS feeder. I use NewsBlur, but there are some other ones. Basically, you can set up these different feeds with different key terms, as well as feeds from different journals that will then give you kind of updates about what's what, but you don't get all these dingy alerts. Instead, they get kind of collated into this one central location that you can then look and see what's potentially of interest to you without having to dig through all the stuff that's not of interest to you. However, sometimes those will miss things, and there are things that are of interest to you, but are not of your typical, the type of things that you're normally interested in, but there's just some really cool articles that come out sometimes, and it can be really interesting to read things from other fields or disciplines and that sort of thing. So I regularly check the online now sections of websites for key journals of interest. I kind of make this my lunchtime habit is to search through, look at my RSS feed, has anything come up? Check the online now sections of websites. So basically of like the journals, Nature, Cell, various molecular cell, all those various ones that Journal of Biological Chemistry, Nucleic Acid Sciences, various ones where I know that things that I'm interested in often get published. And the online now is basically before it gets actually printed, or sometimes they don't even print it, but before it's like actually in the issue. These kind of like pre-access. They're peer reviewed, so they're not preprints, but they're often not like totally formatted and things yet. But yeah, so I regularly check those. So I do that like over lunch. Also, it's nice to set aside like a dedicated period of time each day to do a little reading. So I like to set aside like an hour in the morning where I can just kind of like focus on reading papers if I have time. And also to keep track of all those papers, use a reference manager. So something like Mendeley's, Zotero, ReadCube, to save and organize it all. This is going to make it a lot easier even if you don't have time to read the paper, even if you could just save it to your library this way. If you then kind of remember, oh, there was that paper, I think it was that paper that came out, you can kind of go search for it and then you could read it, even if you didn't have time to read it before. This is also going to be really helpful to have these references. Basically, these reference managers are going to help you. They can like integrate into Word and things like this. So when you're writing a paper or you're writing a report, it'll format the citations for you. So that's how you can how I kind of stay organized. And so again, find what works for you and just stick to it, because really having a consistent routine is really, really important in order to find what you need and then succeed.